Thank you. Good morning, everybody. My name is Ian Bulovich. I'm a data scientist in the Computational Health Informatics program at Boston Children's Hospital. Today, I'll be presenting our research on detection and monitoring of potential outcome reporting bias using large language models and its application to FDA-regulated drug trials. So my principal investigators for this work are Florence Bourgeois and Tim Miller, who are both faculty members in the Computational Health Informatics program at Boston Children's Hospital, as well as associate professors in the Department of Pediatrics at Harvard Medical School. So to start off with some background, selective inclusion and selective reporting of clinical trial outcomes can introduce bias, and these biases can lead to treatments that are not as effective as the research suggests, and potentially more harmful as well. So one tool we have to monitor this is ClinicalTrials.gov. ClinicalTrials.gov keeps track of edits that are made to a clinical trial record over time, so we can look back through the records and identify changes that were made between trials' prospectively registered outcomes and the outcomes eventually reported at the end of the trial. So this is a very useful resource, but in a landscape of hundreds of thousands of clinical trial records, it's completely infeasible to manually compare versions for each and every trial. So our objective was to tackle this scaling issue using large language models. So large language models uniquely enable us to analyze massive amounts of text, which holds promise for a wide spectrum of biomedical questions and challenges. And in our case, we can use a large language model to identify changes to primary outcomes over a large cohort of clinical trials. So in this research, we explore using a large language model to estimate changes to primary outcomes recorded in ClinicalTrials.gov at scale. So the large language model that we chose to use was OpenAI's GPT-4o model, which was one of the smartest models available when we were conducting this research at the end of 2024. We didn't do any fine-tuning of the model, but rather we iteratively developed a detailed prompt instructing the model on precisely how to identify the kind of changes we are looking for. So for each trial, the model compared the record at the beginning of the study to the most recent record available in ClinicalTrials.gov. For some trials, the primary outcomes are never edited after registration, but for those that are, we instructed our model to identify 3 different categories of changes. Outcome addition, so adding a new primary outcome that wasn't there before. Outcome removal, which is removing an existing primary outcome. And substantial outcome modifications, which we define as a change to the outcomes measure, measurement procedure, or measurement timeframe. So just to give an example of what some of these changes may look like, here's a comparison of a clinical trial with the initial record on the left and the final record on the right. So this is a modification to the outcome measure. As you can see, second progression-free survival on the left was changed to first progression-free survival on the right. Here's another example where the measure is the same, but the time frame has changed from 6 months on the left to 12 months on the right. So we also consider this to be a modification of the outcome. But it's important to remember that not all changes that are made necessarily constitute a modification. So in this example, changing percent artery dilation to percentage points of change in arterial diameter is not something that we considered a meaningful change. So this is not something that our model would flag as a modification. So to evaluate the performance of our model, we manually labeled and adjudicated changes made to the primary outcomes of 100 clinical trials and evaluated the performance of our model on that labeled data. So the metric that we use to evaluate our model is F1 score, which is on a scale from 0 to 1 and balances precision and recall. So we can see that for the addition category, our model had an F1 score of 0.88, for removal 0.81, and for modification 0.67. So this is a difficult task, and the model's performance is not perfect, but it does achieve fair performance across all 3 of these categories. So the trials that we chose to analyze with our model were interventional trials registered in ClinicalTrials.gov that were studying an FDA-regulated drug. It's also important that every trial was prospectively registered. So if a trial was registered after the registration deadline, then there's no record of the primary outcome before the trial started, so we can't make a comparison. And finally, we only looked at trials that started from 2008 to 2022 to allow for at least 2 years of follow-up. For our statistical analysis, we performed univariate analyses and multivariate logistic regression across a few different variables of interest, namely the trial start year, the therapeutic area of the trial, whether the trial received any funding from industry, whether the trial was randomized, and the number of enrolled participants. So here's what our model found. 27,227 trials met our inclusion criteria, and among those trials, 40% of them had a change to their primary outcomes in at least one of our 3 categories. So 14% of trials added a primary outcome, 11% of trials removed a primary outcome, and 33% of trials had a substantial modification to one or more of their primary outcomes. Also, more than 4% of trials showed all 3 types of changes. One interesting trend that we found in our multivariate analysis is that the prevalence of these changes has decreased significantly over time. So in trials that started from 2008 to 2010, we found primary outcome changes in 82% of these trials. But by 2020 to 2022, we found those changes in only 29% of trials. Another interesting finding from our multivariate analysis was the effect of industry funding. So we found that trials that received funding from industry were significantly more likely to have a change to their primary outcomes compared to those that did not. So here we can see that for trials with industry funding, nearly 50% of them had some change to their primary outcomes, while for not industry funded trials, that number is closer to 30%. So there are a couple important limitations to address with this work. The first being that with regards to our human labeled evaluation data, annotator agreement is never perfectly objective. So this is a difficult labeling task, even for humans, and it's often unclear whether a certain change that's made to a primary outcome is truly a modification or not, and it can end up being a matter of subjective interpretation. The consequence there is that even the human labeled data may not be the perfect ground truth. It's likely pretty close, but there's certainly some gray area. And what that means is that the modification counts reported by our model should be interpreted as estimates rather than exact counts. Another important limitation is that we chose for our model to only compare trials' primary outcomes. So the classification is being made without any additional context from other parts of the registration. And it's possible that with additional context, the model's decisions could be better informed. The model is also not considering secondary outcomes. So one thing that we see happening sometimes, which we discussed a few minutes ago, is that researchers move outcomes between primary and secondary, and that's not something that our model is able to capture. So to recap, we found that large language models let us monitor changes made to clinical trials efficiently and at scale, far beyond what's possible with manual human labeling. We also found that primary outcome changes in clinical trials studying FDA-regulated drugs are common, they have decreased over time, and they're associated with industry funding. And there are a few future directions we see for this work. One is to extend our analysis to other clinical trial cohorts beyond just FDA-regulated drugs. Also to use newer and more powerful large language models because the capabilities of these models are getting better every day. We're also curious about using similar methods to compare trial registrations to eventual published articles and exploring the differences that appear in the outcomes reported there. And finally, we're interested in applying these large language model driven methods to develop real-time systems to monitor research integrity. Thank you.
Okay. A rush to the microphones. So we'll start in the back there, white shirt, say your name clearly.
Hi, thanks for the very interesting talk. My name is Johannes from University Hospital in Basel, Switzerland. I was wondering when doing the human labeling, I might have missed it. Did you extract data in duplicate and then compare the human extractions to sort of get some inter-rater reliability estimates, which could have sort of allowed for quantification of the difficulty of the task? And my second question is, did you include examples of difficult cases in the prompt? So were these zero-shot prompts or few-shot prompts?
Yeah, thank you. Those are great questions. So to address your first question, we had 3 annotators annotating our 100 manually labeled trials. And then we didn't see perfect agreement across all 3 of our categories, but then we did another pass of adjudication where we had all 3 annotators sort of collaborate to reach a consensus on all of our labels. For your second question, sorry, could you repeat the second question?
Second question was about difficult examples being included in,
Ah.
or like theoretical examples being included in the prompt and what the model should decide then.
Yeah, of course. So our prompt was a few shot prompt. So we did include some difficult examples in the prompt to give the model sort of a sense of what kind of classifications to make. And we found that that kind of prompting did have a significant impact on the model's performance. Thank you.
Hey, Dr. Wee in the front.
Christina Wee, Senior Deputy Editor at Annals of Internal Medicine. Thank you for this paper. As part of our editorial process, ideally, the editor should be adjudicating discrepancies between a registered protocol and what's reported in the published article. So I was trying to understand, who do you see as the end user of these models? Is it for monitoring purposes, by research, or are you anticipating that the individual reader would use this as their, I'm curating, just trying to understand the rationale.
Yeah, thank you. That's a great question, and maybe slightly above my pay grade. But I think one area where this could be helpful is if it were sort of embedded in ClinicalTrials.gov for the researchers as they're entering this information. A lot of the changes that we see don't seem to be intentionally misleading or malicious, but it might be helpful for these researchers if they're making a change, and maybe our model could say, hey, it looks like you're making a significant change from what you reported earlier. Maybe that would help researchers understand like that these differences, they shouldn't be appearing. Thank you.
Okay, I think we'll have time for everybody who's up, so we'll go right here.
Owen Joe, Research Associate at McMaster University. I have a question regarding the stochastic output nature of large language models, which typically is a concern in terms of the reproducibility of your results. I'm wondering if you have done anything to address this, for instance, by repeating the same prompt multiple times and taking an average of your results, or decreasing the temperature to 0 so that the output more or less is deterministic.
Yeah, thank you. That's a great question. So in this sort of more exploratory work, we didn't directly quantify the confidence of our model, but that's something that we're looking at in our continued work on this. And we have a method to sort of put a confidence interval on the model's estimates. So what that tends to look like in sort of what we're starting to see is for the addition and removal categories where the model's agreement was with the human annotators was higher, those confidence intervals look something like plus or minus 5%. And for the modification category, where it's a little bit less aligned with the humans, it's more plus or minus 10, 15%. Yeah.
Thank you for your answer. I just have one more thing. Will you make your prompts available for replication by other researchers?
Yeah, absolutely. So our prompts are available. There's, I believe, a link in the program to a GitHub repository with our prompts.
Thank you.
Yeah, thank you.
Back microphone.
Kyungwon Hong from National Institute of Health / ClinicalTrials.gov. So huge kudos to your research and presentation. 2 questions, maybe just for a clarification. Was your model capturing / reviewing all the historical pages within study record as well, or just 2, I guess, time points, like at the beginning versus at the end? Because ClinicalTrials.gov provides historical pages as well.
Yeah, so our model is only comparing 2 versions of the trial. The first one being the last version that was before the registration deadline. And then we're comparing that to the most recent version available in ClinicalTrials.gov.
Okay, thanks for the clarification. I mean, the reason why I'm bringing this up is I would really love to know, I guess, the intensity of, I guess, the outcome changings within the study level, like how many outcome changings happen within one study. That maybe just for a suggestion. Second question, were you thinking about like subgroup analysis of like pivotal versus supportive or study phases?
Sorry, could you repeat that?
Like, were you considering like, subgroup analysis between pivotal trial versus non-pivotal / supportive trials, FDA?
Yeah, that's a good question. That's not something that we've looked at this point, but yeah, that's something that would be interesting to look at in the future.
Thank you.
Thank you.
Eric?
Eric Turner with Oregon Health and Science University and former FDA reviewer. The primary outcome changes are, of course, raise one's, interest and is whether there's going to be publication bias, but they could all be absolutely, and I think you said this, perfectly legitimate. And the key is, was the blind broken? And then, then things were changed from secondary to primary or vice versa. So one way you might consider, investigating that is actually to look at the FDA reviews also on the ones where there's any suspicion of primary outcome changes. Then you get the FDA reviews downloadable from the FDA website, and then compare that to the final publication. Because if you see it in the FDA review and that's what the FDA analyzed, then even if it's claimed that the primary outcome has changed, you have good reason to suspect that may not be entirely on the up and up. So, and by the way, the association with industry funding could be absolutely legitimate just because pharma's probably has their act together about, being on top of ClinicalTrials.gov and making those changes as compared to investigators with universities that may not have such an affiliation. Anyway.
Yeah, thank you. Thank you for the suggestion. I think it would be really interesting to look at the FDA comments. Yeah.
Robbie in the back.
Robert Thibault, Coalition for Aligning Science. Thanks for the presentation. That's really nice to see a tool that's doing something functional. I'm curious what you think you're practically capturing in these differences. And just a little bit of background context. I led a meta-analysis that looked at 89 studies comparing selective outcome reporting between the registry and the publication. And in total, I looked at over 6,000 studies. And that's capturing something slightly different than you are between the two study registries. We found the opposite association with industry funding, where there's fewer discrepancies from industry funded studies. And we found no difference over time as well. And so we're capturing different things, but I'm curious, if you can speak more to that.
Yeah, that's a really good question, really interesting. So we've considered comparing to published articles as well, but to do that with large language models in sort of an automated way at scale is a much harder task because you're comparing data that's in two different formats, and that leads to all sorts of challenges. I guess our intuition is that if a change is made in ClinicalTrials.gov, then whatever's in the final version of the registry on ClinicalTrials.gov would at least be similar to what is in the eventual published article. But yeah, that's definitely something that we're interested in looking at more deeply, is the differences with published articles. Thank you.
This mic.
Really nice talk. Randy Ellis from Harvard Medical School. So precision, sensitivity, and F1 score are all threshold-based metrics as opposed to threshold-free metrics like the area under the ROC curve. So I'm wondering, how did you pick your decision threshold? What did specificity look like? And what did your AU ROC look like?
That's a really good question and an important point. So one of the challenges with these large language model-based methods is that we're building a classifier that doesn't give us like a number between 0 and 1 as its classification. It's just giving us a binary, this modification is here, modification is not. So we're not actually able to generate an ROC curve from that. But yeah, that's something that our lab is investigating methods to generate that kind of ROC curve.
Thank you.
Thank you.
Okay, and speaking of mics, we have Mike Berkowitz
Yeah.
from representing the whole internet.
Yes, the virtual chat. Juna Shiro from Mayo Clinic asks a question that you've gotten at a bit in some of the Q&A. Protocols can change for legitimate reasons. For example, patients not willing to be randomized to a treatment they don't want. Do you have a general sense of the rationale for these changes? Some may be less on the up and up, and some may be legitimate.
Yeah, that's a really good question. And it's sometimes hard to tell just looking at the registration of the clinical trial. A lot of the modifications that we see are modifications to the time frame. And then we are also seeing a lot of situations where the original version of the primary outcomes were just very underspecified. So maybe not quite so extreme as the researcher just putting in sort of safety as the primary outcome, but something very underspecified where later on they end up having to add more detail. So that's the kind of issue that I think we can probably do better on and avoid. But it's true that we can't really capture the intent behind all of the modifications that we're seeing.
And when you say we can do better on, is that on the researchers, the registrants part, or on your, as the analyst part?
I think on the, probably both. I think it would be nice if we had sort of stricter, more rigorous guidelines for researchers who are registering their trial about how much detail they should be including regarding their primary outcomes.
Great. We have CT.gov in the room, so that's a great suggestion.
Well, I'll ask the last two questions. Unless I missed it, the decrease, I just want to comment on these primary outcome, these trends that you note on the last slide. It was a huge decrease over time. So I'd be interested in your comment on your thoughts on why that was so dramatic. And second, the association with industry funding. Is that good? Is it bad? Is it neutral? I know that industry often, which is very concerned with FDA approval, is much more attentive to making sure the record is accurate, and academics sometimes don't update that record quite as conscientiously.
Yeah, those are both great questions. So for the decrease over time, I can't say for sure what has caused that, but potential causes could be just increased awareness about issues with outcome switching, and then also maybe improvements in the internal quality control process in ClinicalTrials.gov. But that's definitely something we want to look at more closely. And for the association with industry funding, I'm really not sure. And I'm sure there are people in this room who could speak to that better than I can, but yeah.
Okay. Well, let's thank Dr. Bulovich for a great presentation and a very full discussion.
Thank you.